Hi everyone, I'm Shalini Gupta. I have done my graduation in life sciences from Delhi University and my master's in biotechnology from South Asian University. Currently, I'm pursuing my PhD in biotechnology from Jawaharlal Nehru University. As I have been in research from so many years, it gives me confidence to have deep understanding about my subject that I would like to share with my students. And I also believe that I have ability, skills, and attitude to teach my students and explain the concept with more clarity. Now I'll give a quick glance about the subject. So first unit is diversity in the living world, where we'll discuss about the living world system, how the classification is divided, like what are kingdoms, how they are divided into phylum, what are the subdivisions, and in chapter 2 we'll discuss more about biological classification, where we'll talk about algae, then plant system, then animal system, five kingdom system, six kingdom system, and then we'll come on to the plant kingdom where we'll talk about different phylum like algae, bryophytes, pteridophytes, gymnosperm, angiosperm. And in animal kingdom, we'll talk about porifera, coelenterata, annelids, phylum insects, and so on. And in second unit, we'll talk about flowering plants, where we'll discuss about the morphology and the anatomy. In morphology, we'll talk about leaves, roots, shoots. In anatomy, we'll talk about the tissue system, complex tissue system, simple tissue system. We'll talk more about the xylem and phloem. In structural organization in animals, we'll first talk about tissue system, then we'll discuss about cockroach, frog, and their dissection. Then we'll come to the third unit where we'll discuss about cell in detail. First, we'll talk about what is cell, who has given their this theory, what are plasma membrane, what are organelles. Then we'll move on to the biomolecules where we'll talk more about carbohydrates, lipids, and proteins that we have read somehow in 10th class, but now we'll discuss in more detail. Then we'll move on to cell cycle and cell division. Here we'll talk about mitosis, meiosis, what are G naught phase, what are S phase. Then we'll move to the next slide where we'll talk about plant physiology and human physiology. These two are my favorite unit, and second point is they also have more marks. They also grab more marks, and it's very interesting to read about them. First, in chapter 11, we'll talk about transport in plant, where we're going to discuss about how food are transported from shoot to root and how water is transported from root to shoot, and so on. We'll discuss about what is transpiration, guttation, and so on. In mineral, we'll talk about macronutrients, their deficiency in photosynthesis. We'll read about light reaction. This is my favorite topic. Light reaction, dark reaction, C3 cycle, C4 cycle, and in respiration we'll talk more about glycolysis that we know about something about it. Glycolysis, Krebs cycle, etc. How much energy ATP is utilized, all these things in respiration. It's very important chapter. Then we'll talk about hormones of plant. Then we'll move on to the human physiology where we'll discuss about digestive system, and in breathing and exchange of gases we'll talk about respiratory system, lungs, and in circulation we'll talk about heart, circulatory system. In excretory products and elimination we'll discuss more about kidney, nephrons, locomotion and movement. We'll talk about muscle contraction thing. Neural control and coordination. Here we'll talk about brain. It so fascinate me so much. Chemical coordination and integration. Here we're going to talk about hormones of humans. What is dopamine? What is oxytocin? And so on. Then, in next unit, we'll talk more about sexual reproduction, asexual reproduction. Then we'll talk about gametogenesis in flowering plants, sexual reproduction in flowering plant, what are the ways to do it. And human reproduction, we'll talk about what are male reproductive part, what are female reproductive part. And in reproductive health, we'll talk about different kind of diseases also. In genetic and evolution, in chapter number 5, we'll discuss more about Mendel theory. First law, law of segregation, law of dominance, law of codominance, law of incomplete dominance, and so on. Then in molecular basis of inheritance, we'll talk about DNA. It to fascinate me so much. Protein. आह वाओ, टाइप्स ऑफ डीएनए दैट एग्जिस्ट, ट्रांसक्रिप्शन क्या होता है, translation क्या होता है and replication क्या होता है. We'll get to know more about it, and then we'll move on to evolution where we'll see how life originated, what were the ways, what were the way. Next, we'll talk about biology and human welfare. We'll discuss about the diseases. ठीक है? Communicable, non-communicable, and in this strategies, we'll discuss about plant breeding system and so on. In microbes, we'll discuss about what are good microbes for human and what are bad microbes for human. In biotechnology, we'll discuss about cloning, what are the methods to enhance the production of food, production of protein. So one of the topic I'm going to teach here today. And in ecology we'll discuss about how the environment is balanced, how the ecosystem chain works, what are predation, what are amensalism, what are commensalism, what is biodiversity, and what are the issues that we are facing these days, what is Montreal Protocol, what is Kyoto Protocol. All these things we'll study in this unit. Now I'm going to teach here a short topic, that is how to construct recombinant DNA, and one of the tool I'm going to teach here. So first, there are three basic steps to construct recombinant DNA. First is identification of your desired gene, because you want to आप इस desired gene को express करना चाहते हैं अपने mammalian या bacterial cell में. ममेलियन सेल में आप तब एक्सप्रेस करते हैं जब आपको एक्सपेरिमेंट सेल्स के अंदर करने होते हैं. बैक्टीरिया में आप एक्सप्रेस करते हैं या तो आप लिपिड आइसोलेट करना चाहते हैं या तो आप प्रोटीन आइसोलेट करना चाहते हैं, बट विल नॉट गेट इनटू दैट डिटेल. ठीक है? सो आपने अपनी डिजायर्ड जीन को आइडेंटिफाई किया, आपने उसे वेक्टर में इंट्रोड्यूस किया. कैसे? दैट विल रीड अबाउट इट. ठीक है? एंड देन जब आपकी वो लाइगेशन हो गया वेक्टर का, आपके डिजायर्ड जीन का वेक्टर में, तब आपने उसे ट्रांसफॉर्म किया. आपने उसे ट्रांसफॉर्म या ट्रांसफेक्ट किया बैक्टीरियल या मेमेलियन सेल्स में रिस्पेक्टिवली. सो देयर आर टूल्स टू क्रिएट रिकॉम्बिनेंट डीएनए. इस रिकॉम्बिनेंट डीएनए को क्रिएट करने के प्रोसेस को हम क्लोनिंग कहते हैं. ठीक है? So there are so many tools like restriction enzymes, polymerase enzyme, ligases, vectors and host organisms. और इन tools का अपना अपना काम है. ठीक है? Right now I'm going to teach about restriction enzymes only. So restriction enzyme क्या करता है? आपके डीएनए को स्कैन करता है और जहाँ पे भी उसकी साइट प्रेजेंट होती है उस पर्टिकुलर रिस्ट्रिक्शन एंजाइम की, तो वो वहां पे कट कर देता है. और कट करने के भी अलग अलग प्रोसेस होते हैं that I am not discussing here because it has not been given in your syllabus. So there are two types of restriction enzyme, exonuclease या endonuclease. Exonuclease जो होगा वो आपके अगर ये आपका डीएनए है, एक मिनट, अगर ये आपका डीएनए है तो डीएनए के जो कॉर्नर पे कट करें वो आपका एक्सोन्यूक्लियस है. बट एंडोन्यूक्लियस भी थ्री टाइप्स के होते हैं. ठीक है? टाइप 1, टाइप 2, टाइप 3. राइट नाउ आई एम टॉकिंग अबाउट टाइप 2 हेयर. ओके? सो ये बीच में कट करेगा, बट ये अपनी ही साइट पे कट करेगा. एक रेस्ट्रिक्शन एंजाइम ऐसा भी होता है जो जहां पे उसकी साइट होती है उससे थोड़ा दूर जाके कट करता है. ठीक है? So the one that is required in cloning is the type 2 restriction enzyme. Okay. Okay. So now I am going to show you a schematic representation of type 2, how type 2 endonuclease work. That has been shown in your NCERT also. So ये आपका वेक्टर डीएनए है, ये आपका डिजायर्ड जीन है जिसे आप अपने वेक्टर में इंट्रोड्यूस कराना चाहते हैं. तो ये कुछ ऐसा बन जाता है आफ्टर क्लोनिंग, जो मैं आपको दिखाना चाहती हूँ एक बार. सी फिगर उतनी अच्छी नहीं बनी है, बट ये जो रेड है ये वेक्टर है और जो ये येलो है दैट रिप्रेजेंट्स योर डिजायर्ड जीन. फिर उसको आप अपने बैक्टीरिया में डाल देते हैं, ट्रांसफॉर्म कर देते हैं, जिससे आपके पास नंबर ऑफ जो डीएनए है उसका अमाउंट बढ़ जाता है. कैसे बढ़ जाता है? आप उसे ओवरनाइट छोड़ते हैं ग्रो करने के लिए, बट अभी हम वहां तक नहीं जाएंगे. पहले हम एक सिंपल रिस्ट्रिक्शन एंजाइम समझते हैं कि काम कैसे करता है. तो वेक्टर डीएनए लिया, वेक्टर डीएनए पे रेस्ट्रिक्शन एंजाइम ने कट किया, वो इको आर1, ये एक एग्जांपल दिया हुआ है, इको आर1 इसकी जगह पे हिंड 3 भी हो सकता है, एनसीओ1 भी हो सकते हैं और भी होते हैं. ठीक है? इसने यहाँ पे कट किया, इसने इसको आइडेंटिफाई किया, फॉरेन डीएनए में भी इसने आइडेंटिफाई किया, तो ऐसा स्टिकी एंड क्रिएट कर दिया. तो वेक्टर डीएनए का स्टिकी एंड जाके आपके फॉरेन जीन के स्टिकी एंड से लाइगेट कर दिया बाय डीएनए लाइगेज। तो आपके पास आ गया आपका एक रिकॉम्बिनेंट डीएनए। अब इस रिकॉम्बिनेंट डीएनए को आप आगे ट्रांसफॉर्म करेंगे अपने बैक्टीरिया में, ठीक है, और फिर उसे ओवरनाइट ग्रो करने के लिए छोड़ देते हैं। आज के लिए इतना ही, ठीक है, बाकी हम नेक्स्ट क्लास में पढ़ेंगे। थैंक यू सो मच।